All right, welcome to another glorious day of biology. Today we're working on our Osmosis and Diffusion Lab. So either pull this out, this paper copy, or on line and from your Google Doc. First things first, I want you to take a look at the important background. I want you to read through this, things like passive transport, diffusion, facilitated diffusion, osmosis, and active transport. I want you to go through that, hit pause right now, read through that, and then hit play once you're done. All right. The problem today, how can you determine whether solutes are diffusing across the membrane? Introduction here. So a cell membrane is a selectively permeable barrier. Some particles can pass through the cell membrane while other particles are held back. Solutes that can move across the membrane generally do so by diffusion. When solutes diffuse, they move from an area of high concentration to an area of low concentration. Diffusion of certain solutes can be facilitated by protein channels that act like tunnels through the cell membrane. In this lab, you will use dialysis tubing to represent the cell membrane. Dialysis tubing is a synthetic membrane used to filter waste from the blood of a person who has experienced kidney failure. The tubing has small openings, or pores, that allow the passage of relatively small molecules. You will use indicators to determine which molecules diffuse through the pores. So the materials we have here, materials list. Safety is first, so make sure you have your goggles on. So we're going to read pre-lab questions here. It says, read carefully the procedure for data collection below and answer these questions. So let's take a look at that procedure. So the procedure says you will determine if the analysis membrane is permeable to starch, iodine, and or glucose. Testing permeability to starch and iodine. Number one, put on your goggles and get about 15 centimeters of dialysis tubing. This is the dialysis tubing right here. Add tap water to a plastic cup to make it 3/4 full. Number three, soak the tubing in the running tap water for 1 minute. Number four, gently roll the upper end of the tubing between your fingers until it opens. Number four, A, I guess. Double four, maybe. Remove the tubing from the water. Fold up 1 centimeter of the tubing at one end. Use a clip to tightly seal off the folded end. We'll skip, that's supposed to be go to 6 now. Right here. It says pour about 10 milliliters of starch solution into the tubing. Number 7, add about 10 milliliters of glucose solution into the tubing with the starch solution. Number 8, fold down 1 centimeter of the tubing at the open end. Use a second clip to seal this end. Number 9, use tap water to thoroughly rinse the outside of the tubing. 10. Gently pat the tubing dry. Weigh the filled tubing and record the mass. 11. Place the tubing in the cup of tap water. It must be completely underwater. 12. Add about 20 drops of iodine solution to the water in the plastic cup. Record your initial observations in data table 1. 13. Wait at least 15 minutes or overnight and complete the while you wait questions. 14. Use a glucose test strip to test the liquid in the cup outside the tubing and record the result. 15. Gently remove the dialysis tubing from the cup and record your final color observations. Gently pat dry the tubing, weigh and record the mass. So let's take a look now at the pre-lab questions here. Number 1 says, what color is iodine in plain water? What is iodine's color in water? Think back to the carbohydrate lab. What happens when iodine and starch combine? If you don't recall, go back to the carbohydrate identification lab. Watch that video. Number 2, where does the starch start? As in, like, where did you initially place starch? How will you know whether starch has diffused across the membrane? Now remember, this is going back to where is starch placed? Where is the iodine? What happens when the two combine? So how will you know if starch moves across the tubing? Number 3, where does the iodine start? So where did you originally put iodine? How will you know whether iodine has diffused across the membrane? Once again, where is starch located initially? What happens when you combine starch and iodine? Number 4, where does the glucose start? Where did you initially put glucose? How will you know whether glucose will diffuse across the membrane? So take a look at the directions. And how are we going to test if glucose is present? Number 5 here, use analogies. How is a window screen similar to a cell membrane? What's the function? What is a cell membrane? What's its function? And how is a window screen different from a cell membrane? So think about that. Next, let's start this lab here. So first part here, it says put your goggles on. We're going to add water to a cup. So we have our cup of water right here. It says soak dialysis tubing in water. So I don't have a sink right next to me, so we're going to use this beaker of water. So if we take a look here, here's our dialysis tubing here. It's kind of crinkly. So we're going to dip it into the water. Wait for a couple seconds. Take it out, and all of a sudden, it's very malleable. Then it says roll it in between your fingers. I don't know if you can see this. Okay, stop moving. See, it has started to open up now. Now it says take a clip, so we're going to take it. I take the other end because this is open. We're going to flip about a centimeter or so of it. I'm going to take our clip here. I'm going to clamp it down so you have a nice good seal. Now says add starch. So this is going to be my spill. In case I spill some, it's not going to go all over the table. So I have our test tube in here. Let's get that a little bit, dapping it down here. So rub it back and forth. And make sure it opens. So we have our starch solution. Place it in and add. And so we're going to kind of spitball it a little bit here, about 2 fingers lengths. So now you can see here we go. We've added starch. Now it asks for glucose. Add some glucose here. Now it says at 8, fold down 1 centimeter. Take a clip. Clip it so that nothing can escape. Now use tap water to rinse. Now we're going to rinse this off. So we're going to rinse this off by bringing in some tap water. Now asks us to take and dry. Gently pat the tubing dry. Weigh your test tube. Weigh your tubes. We're going to take it. We're going to dry it off here. Pat it nice and dry. So now it's nice and dry. We're now going to find the mass. Place it on here. So we have 25.9 grams, so 25.9 grams. So now we're going to take on in the chart that we have here, where it says initial mass right there, we're going to put in our 25.9 grams. Now we're going to move this guy over. Bed of truth. Now, here's our cup of water. There we go. Sure it's underneath there. Okay. And now it says we've patted it down. I'll place the tubing in a cup of tap water. It must be completely under. So we're going to take our tubing, place it in the tap water so that it submerges. Add just a little bit more. There we go. Add a little more tap water. So now it should be completely submerged. Excellent. Now it says add about 20 drops of iodine solution in the water in the plastic cup. Record initial observations in data table 1. So we'll add about 20 drops here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 18, 19, and 20. Okay, now it says wait for 15 minutes. But let's take a quick here. Let's fill in the chart here. So, what was the initial color of the tube? That was, oops, did I drop water all over the place? That's clear. Was glucose present in the tube? Yes. Was starch present in the tubing? Yes. Did we put any iodine in the tubing? No. Now, let's take the color of the cup. Nice background here. What is that color? Yes, I agree, it is yellow. Was there any glucose in the cup? No. Is there any starch in the cup? Well, if there was starch in the cup with the iodine, what color would it be? Yeah, it'd be dark blue or purple. Is there iodine in the cup? Yes. All right, so now we wait. While you're waiting, hit pause and go to the while you are waiting questions. Answer which molecule is larger, glucose or starch. Think about them. Which one's a monosaccharide? Which one's the polysaccharide? If you don't remember, go back to your carbohydrate section of your notes. 2, diagram, basically draw a diagram of the cup and tubing and what's located where. And then use arrows to hypothesize what you think will happen during this experiment. So what's going to go in, what's going to diffuse. And then 3, why did we add iodine to the water in the cup, but not in the dialysis tubing? Explain your answer thoroughly. Pause, answer the questions, and I'll have to do some splicing. We'll see your results in just a bit. Here we go. Let's take a look. It's been about 25 minutes or so since we last talked. So first off, let's take this out. Has the tubing changed? Well, you tell me. Has it changed? Yes, it has. So, what color is it now? Is glucose present? Well, what do we know about diffusion? Diffusion or osmosis will happen until what takes place? It'll continue to happen until they're equal. So, if we had glucose in the beginning, should I have glucose in there now? Yes. Is starch present? Well, what do we know about iodine and starch? Yep, so therefore we know that there is starch present in there, and we know that iodine is present as well because it turned the starch color. Now let's take a look at the tube, or the cup. Looking at the cup here, it says, what color is it? All right, is glucose present? Well, to do that, now we take one of these strips right here. Here's, oops, there we go. Glucose test strip. Take it, place it into the cup. If it turns green, if it darkens and turns green, then there's glucose present. So with the tap water, there was none, but now, let's take a look. Sure enough, there's now glucose present in the cup. So what does that tell you about where was the glucose originally? And now where is it? So did it diffuse? What does it tell you about the size of the glucose? Just remember that this dialysis tubing is semi-permeable. Is starch present in the cup? Well, we have iodine in the cup. What do we know about iodine and starch? And when they combine? And of course, we know that iodine's in the cup because it's yellow. Now, it says, let's take a look at finding the mass. Let's try to move our scale here. I'm going to measure it now. What does that say? It says, oh, got some water there. All right, our mass is now 27.3. 27.3. All right, so we added some iodine. Is the iodine enough? Oh, I got water there now. Is the iodine that we added, that entered into this dialysis tubing, is that enough to make up for that much of a difference in mass? No, of course not. So what would have then moved into, or what could have then moved into the dialysis tubing to increase its mass? All right, so make sure that you write the mass in the appropriate spot. All right, now is the time to answer the analysis and conclusion questions. Use your data and your knowledge of the size of starch versus glucose and your knowledge of diffusion and osmosis to answer the questions. Good luck.